Hi, my name is Tom Onens. I work at the Chatterjee Lab at the University of Colorado Boulder. I'm going to be talking about how our lab has used antisense inhibitors to take RNA-seq data and use that to resensitize carbapenem-resistant E. coli to carbapenems. Just as a brief background, the antimicrobial resistance crisis has been accelerating in recent decades. You can see the number of deaths per year in the United States and globally up on this slide. Within this crisis, carbapenem-resistant enterobacteria are also a huge issue. They've been ranked as the highest priority by both the World Health Organization and the CDC. So with this study, what I'm going to be talking about is one specific isolate from the University of Colorado Medical Center. It's this CRE E. coli. And it has shown a lot of resistance to a whole bunch of different antibiotics. Notably, we have resistance to ertapenem, not meropenem. So what we thought is that we could use this isolate to kind of study the development of resistance and maybe try to reverse resistance to carbapenems. And the way we do that is with this sort of alternative antibiotic approach that we use in this lab. So they're called peptide nucleic acid. The reason we use them is because they're highly sequence specific. They can target exactly the pathogen you want and leave the commensal bacteria alone, which is important because a lot of broad spectrum antibiotics kind of increase the evolutionary pressure towards resistance by killing everything. And these are kind of peptide nucleic acids are peptide nucleic acid hybrids. And what they do is they bind to the start codon region of an mRNA and they stop progression of the ribosome. So if you target these peptide nucleic acids, as PNA, to an essential mRNA, it won't produce that essential protein. The bacteria will die. And so the question is, with PNA is how do you select the right target and how you design PNA that are going to be most effective while leaving the most other stuff alone. And the way we do that is with this thing called FAST. It stands for Facile Accelerated Specific Therapeutics. Is this platform we use. So it starts out, and this is going to kind of permeate through the talk. We design PNA, and in this study I'm going to design PNA using both genome screening, which is more standard, and then transcriptomics, which hasn't been demonstrated before. We designed the PNA sequences to have minimal off-targets and be soluble based on the sequence of the PNA. We build them using an FMOC peptide synthesizer, which is just a standard peptide synthesis machine, and then we test them. In this case, we're going to be testing PNA in combination with carbapenems. The idea here being that we're not going to target something essential to the bacteria under normal growth. We're going to target something that's essential to the bacteria under carbapenem challenge. And we're using our genome screening and transcriptomics to figure out what that is. So we're taking that CR coli, and the first thing we looked at was beta-lactam related resistance genes. We want to know if there are any resistance factors that would genomically that would confer resistance to these carbapenems. So the first thing we find is a PBP2. That is a substrate for carbapenem action. The ones we're going to be using are ertapenem and meropenem. They both bind to this, and it is a perfect match with the PBP2 from a reference strain of E. coli that is sensitive to both these drugs. So that is not the cause of resistance. We know that off the bat. The second thing we're going to look at is these two beta-lactamases. They're plasmid associated. Neither of these has either been associated with carbapenem resistance, so we're 0 for 2 there. The first interesting thing I find here is something called AMPC2 in the Argonaut database. This is known as the AMPC beta-lactamase in MG1655, the reference strain of E. coli. Basal expression of this, notably, is not enough to confer resistance, but if you overproduce it and there's porin deficiencies, it can contribute to ertapenem resistance. So maybe that's our first hit. That's our first PNA that we're going to design to try to knock down, see if we can restore some sensitivity. Alpha AMC is the first PNA we design. Next, we're going to move on to the main, and that is transcriptomic PNA design. What we do here, we have take an overnight culture of E. coli, we dilute it 1 to 20, and then grow it up for an hour. Then we treat it with sub-mic concentrations of both ertapenem and meropenem. Remember, ertapenem, it is resistant, meropenem is sensitive. And then we compare these to the no treatment condition, and that gives us a whole bunch of differential gene expression. What we do to sort this is we put it into a big Venn diagram to see what is commonly upregulated, what is commonly downregulated. And we pulled out a few interesting targets that we found from this Venn diagram. The first one is FLHC. That one is consistently downregulated in all conditions. That's in that middle 6 there. It is commonly downregulated with a whole bunch of other motility genes. So the motility ones are something we're going to keep an eye on. The other two come from this ertapenem overlap here at this 38, and that left-hand side of the Venn diagram. HYCA and MALT, they are regulators of the HYC and MAL operons, which are all upregulated consistently in these experiments. So we wanted to see what knocking down those regulators does. We did a second transcriptomic experiment, and that is with small RNA sequencing. So this enriches for smaller RNA transcripts just to make sure to cover all our bases, make sure we didn't miss anything in the total RNA sequencing that will pop up here. And what we do find is different differential expression profiles. So we also pull a few genes from this. Notably, we have one in here, YGAC, that can discriminate between the meropenem and ertapenem response. In the past Venn diagram, we didn't see any meropenem overlap genes that weren't also shared by ertapenem. And then DSRB and BOLA we also found might be important. BOLA specifically because it is a regulator of AMP-C, which I mentioned earlier. So that leaves us with 7 different PNA to synthesize. And once we synthesize and purify them, we're going to test them in combination with ertapenem or meropenem, and we expect them to show some sort of interaction with these drugs. And the way that we measure that is through antagonism and synergy. In a typical combination treatment, it's really a multiplicative inhibitory effect, as you can see on this plot here. If the combination experiment grows better than we expect, that is antagonism. If it grows worse than we expect, that is synergy. And we expect different things depending on how these PNA targets performed in the transcriptomic studies. So I don't have time to go through all the results. I'm going to go through the results that I find the most interesting. So the first thing we have is alpha AMP-C, the PNA that's supposed to inhibit the beta-lactamase. And in combination with ertapenem, that behaves exactly as we expect. It inhibits growth 76% more than you'd expect based on the single therapies. And this is something that's been established by literature. It's not going to have an effect on meropenem, which we also see. And people have shown previously that you can design PNA based on genomic data. Now, the interesting thing is when we add our transcriptomically derived PNA, we get similarly good results. Here's an example of alpha DSRB, something that's not associated with carbapenem resistance. We only found it by looking at the transcriptome, and it inhibits growth by 85% relative to the expectation, even more than the AMP-C targeting PNA. We get similar results from BOLA, and we get a significant effect in HYCA targeting PNA as well. Now you'll remember BOLA was also supposed to have an effect when combined with meropenem, and we do get that as well. We see that when you bump the concentration of alpha BOLA up to 15 micromolar, so it just really demonstrates you need to play around with concentration a little bit, but these therapies do work, something we might need to study more in the future. The next really exciting result that we got was with the 2 genes that we found to be downregulated in response to antibiotic challenge. What we wanted to see was if we knock these genes down using PNA, can we restore antibiotic resistance in this strain at inhibitory concentration? And what we find, here's an example with alpha FLHC, the motility gene, is it actually works really, really well. That lower line there, the purple line, is the strain's growth when challenged with 0.25 micrograms per mil meropenem. The blue line there is that same concentration of meropenem, but we also add 10 micromolar of the alpha FLHC PNA, which points to possibly the idea that you can tease out the ways that a cell might start to develop resistance. We see a similar result when you have the alpha YGAC PNA at 10 micromolars, 93% growth recovery there, and neither of these rescues growth in the ertapenem-treated CRE E. coli. So you may have some already activated resistance factor going on there. It could point it in a direction towards resistance. So I think that's really interesting for future study. Overall, we got really good results with PNA, and we kind of opened up some avenues for more study of these genes and more transcriptomic results. The only one that didn't really work was MALT. That one actually wasn't specifically upregulated in response to ertapenem. It was really its operon. So that's an interesting note for the future. But in conclusion, we've shown that transcriptomic data can be used for antisense PNA targeting of these MDR pathogens, particularly useful in these less studied non-annotated pathogens where we don't know what the essential genes are, we just have the RNA-seq data. Motility genes, such as FLHC, we found may be important. There's a whole bunch of other motility genes that we didn't explore, that we didn't inhibit, that were also downregulated. So there's another avenue for exploring resistance development. And we found, very importantly, that CRE pathogens using transcriptomic data can be resensitized to carbapenems even when they possess active beta-lactamases like AMP-C. In the conclusion, I'm going to want to thank my lab, my advisor, Anushree Chatterjee, and my co-author on this, Dr. Keisha Erickson.